Okay, so I want to turn things over to a member of our organizing committee, Rob Dunn. Rob is also on our advisory board of TRISEM, and he's going to introduce the speaker for today. So take it away, Rob.
Thanks, Charlie. It's my great pleasure to introduce Athena Aktipis today. So Athena is now an associate professor as of very recently. Congratulations, Athena, in the Department of Psychology at Arizona State University. And Athena is thinking about cooperation in an evolutionary context in many different facets of our lives. And so whether that's in the context of kombucha, in the context of fireside community engagement, or in the context of cancer. And I think it's safe to say that Athena is one of the world leaders in thinking about cooperation, and especially in thinking about that cooperation in ways that relate back to what people do at home every day. And I find Athena's work very inspirational, and especially Athena's new book, The Cheating Cell. And this is a wonderful book. Order it. As you will see, Athena's a great speaker. Invite Athena to speak at your institution, virtually or someday in person. And I just wanted to quickly read from the back some of the supporting blurbs for this book. And so Athena has blurbs from David Quammen, Daniel Lieberman, Steven Pinker, but this one from Barbara Natterson-Horowitz really stood out, the co-author of Ubiquity. She writes, "The Cheating Cell is an instant classic, a book that will transform how physicians and their patients understand cancer, how investigators develop therapies, and how we as a society can work together to reduce the global burden of this disease. Masterful, powerful, and absolutely essential reading for anyone who truly wants to understand the nature of cancer. The Cheating Cell is a tour de force." And what's amazing about that blurb is I think it's entirely accurate. This is an amazing book. This book will have a big impact, and I think we're really very lucky to have a chance to have Athena with us today. And so without further ado, I'll pass the mic to Athena, but I really look forward to this, and I look forward to everything that will come from this book and from your work. So thanks for joining us today, Athena.
Thank you so much for having me. It's a real pleasure. Thanks, Rob. Thanks, Charlie. Thank you, Meredith, for helping to organize. I'm really excited to have a chat with all of you about my book. And I want to invite you all to ask questions as I go. I have slides, but the point of this really is for us to be in conversation. So if you have questions, or if there are things that you want to talk about that are related to, if you've read the book and you have other questions, I would love to have conversations about all those things. So I'm going to go ahead and start sharing my screen here, if I can figure out where my cursor is. Having a technical problem here. Okay, here we go. Let me share here. All right. So just to begin, I want to let all of you guys know, if you want to order the book, you can get a discount if you order it through Princeton. And there's a little code here. I'll put that at the end also, so that you can get that. But I also want to encourage you, if you have a local independent bookseller, go there first, because local booksellers, independent booksellers struggle as it is. And now with the pandemic, it is even harder for them to be, making a living. So check your independent bookseller first, and then you can try Princeton next if you want, and you can get that discount. Okay. So what? Sorry, I'm having some technical problems. Let me make sure that this is sharing properly. I'm gonna have to unshare, I think, and then reshare. I'm not sure. Oh, there it goes. Okay, here we go. So yeah, we'll be having a discussion today about my work, my book, and broader work that is happening around evolution and cancer. So this is a picture of what is called a crested cactus. It is a cactus that has mutations in the growth tips that make it so that it doesn't grow in the normal fashion. And I wanted to present this to all of you because I think it's a really interesting example of how cancer-like phenomena happen across the tree of life. So, you know, it's not just humans, it's not just mammals, it's not just animals. We see cancer in essentially all forms of multicellularity. Now, there's some debate about what we should or shouldn't call cancer. Some people might argue that we shouldn't call these cancer-like forms on plants cancer. The technical term for them is fasciations, and they basically happen when the growth tip, the meristem, which is sort of like the stem cells of plants, rather than kind of being in a tip, they end up growing into a band, which then starts to sort of proliferate and then can create all of these amazing forms. So this photo is actually from the crested cactus garden at ASU that me and a few of my collaborators started. And the reason that we decided to put together this garden is because we thought that it would be a way of talking about cancer that both sort of grounds it in the evolution of life, and also can potentially be an inspiring space for people who have struggled with cancer themselves or have had family members who have struggled with cancer, because it really shows how cancer is a part of life and how these plants can and do live with cancer. So I'll come back to plants with cancer in a little bit, but I wanted to set the stage with this because I think it's a really powerful and beautiful example of cancer that can help us wrap our minds around it in a deeper way. My slides are not moving the way they should.
Athena, click on the actual image. Sometimes I've had this happen.
Ah, there it is.
You have to activate the screen.
Thank you. Okay, so this cactus garden was a collaborative effort, not just among scientists, but also with the grounds crew at ASU and with a master gardener in the UK. So we, you know, put this together, and I wanted to bring this up also because one of our goals with the Arizona Cancer Evolution Center and with our work more broadly is to engage with the general public, to engage in interdisciplinary ways, and to create these sort of surprising collaborations that move us in new directions. So now, you know, we have this amazing garden at ASU, and we also have relationships with people at ASU that we would not have otherwise met and gotten to know because of that. So here's a few other images from the garden. So one of the things that we added because of our collaboration with an artist, Pamela Winfrey, is these tags you'll see on the bottom on the right. And these are opportunities for people to write the names of people who have been impacted by cancer and then put them on these trees that are in the garden to help shade the cacti. And so it's a way for people to participate as well. So all of this is to set the stage now for talking about what we can all do to understand cancer and to change our perspective. And what I want to argue is that humans, the way that people have thought about cancer is often in a sort of human exceptionalism kind of way, that, you know, cancer is a human disease, it's a modern disease, and it's something that we really just need to sort of focus on humans in order to understand it and in order to solve the problem of cancer. But the fact is that we're not alone in our struggle with cancer. It's something that has affected life since the origins of multicellularity. So the reason that I'm studying cancer and how I got into it is because I have been intrigued by the problem of how you create and maintain large-scale cooperation. And so I work across systems, everything from human societies to, Rob mentioned kombucha and cooperation among microbes in kombucha, and of course, looking at multicellular body as a cooperative system. We have 30 trillion cells, they're coordinating and cooperating to make us function. But like with any cooperative system, they face this problem, this challenge of cheating from within. And so I became interested in the problem of cancer because it was a very large scale version of this problem of cooperation. So for my PhD, I looked at computational models of the evolution of cooperation. And then for my postdoc, I ended up at the University of Arizona in the Ecology and Evolutionary Biology department, where I started studying the evolution of cooperation and cancer evolution with John Pepper. And that's when I realized that there was this deep parallel. And also, it's when I first started seeing, probably out of the corner of my eye, these beautiful crested cacti, because they are several of them in Tucson. And I started becoming intrigued by, what cancer looks like across different species as well. Carlo Meli and I then shortly thereafter moved to UCSF and started the Center for Evolution and Cancer, where we started building the community looking at evolution and cancer and holding meetings. And then we went to the Wissenschaftskolleg, where we were part of an awesome working group on cancer evolution. And it was in this working group that we started looking in depth at how cancer manifests across life. And I'll show you some photos of cancer and cancer-like forms across life in just a few slides here. All right, so now I'm at ASU, where we have the Cancer Evolution Center, the Arizona Cancer Evolution Center, where we're looking at all these different levels of how evolution applies to cancer. So how cancer cells evolve in the body. We're looking across species at cancer, and we're doing a lot of efforts for outreach and education. My book is both sort of meant to be a book for the scientific community, but also one that is accessible to more general audiences if they want to engage with it as well. All right, so let's go back for a moment to the origins of multicellularity, so we can really see how cancer plays this role in sort of affecting the viability of multicellular cooperation. So we can think of multicellular bodies as essentially cooperation incarnate. All of us are made of cells that exhibit these abilities to inhibit their proliferation, to control cell death, to actually allocate resources effectively among cells, to divide labor, right? You have different cell types doing different things, and then also to create and maintain the extracellular environment. This is something that's often kind of not talked about, but I think it's extremely important that, yes, we are made of cells, but we're also made of all of the things that are in between our cells and among our cells that help us function. And our cells create all of those things that are in between the cells that help to support our functionality. And creating those things and maintaining them is work. And there is definitely a parallel there between if humans or other organisms are sort of creating a niche, creating an environment in which they can survive and thrive, our cells need to do the same thing. And essentially what happened during the transition to multicellularity is that cells evolved all of these capacities for cooperation. And now that didn't necessarily strictly happen, right when multicellularity evolved. There were a lot of cellular capacities that were in place before the transition to multicellularity, just when cells were sort of evolving to be social, before they sort of decided, I'm putting in scare quotes here, to sort of work together as a unit and reproduce as a unit. So even before that, there were a lot of pieces in place. But then with that transition to, reproducing as a whole organism, it was essential that cells were able to regulate many, many aspects of their behavior in order to cooperate effectively. And essentially what happens is that in cancer, you get a breakdown of that cellular cooperation. So we can look at this now as an exact parallel here in terms of, here are the foundations of multicellular cooperation. These are the 5 foundations that are extremely important for multicellularity to work. And what happens in cancer is a breakdown of all of those aspects of multicellular cooperation. So cancer really is cheating in those foundations of multicellularity. Now, some of you who are into the evolution of cooperation theory might be wondering at this point, well, what do I really mean when I say cheater? Do I mean that there's something intentional going on? What is cheating? Well, for the purposes of sort of thinking about what's going on in multicellular bodies, and largely in general, when we think about cheating, you don't need for there to be any consciousness behind that. It doesn't have to be an intentional act. If you have shared rules that enforce cooperation, and then you have some entities that are breaking those rules and getting a fitness advantage, then that's one definition of a cheater. And that's the definition of a cheater that I use here. And one other thing I just want to mention is that in the context of multicellularity and cancer as a breakdown of that, the shared rules here are actually the genes that are coding for those cooperative cellular behaviors. And if you get mutations in those genes, then you get a breakdown of those cooperative behaviors, and you can get the emergence of cheating. So all of us know that when you have mutations, right, that can lead to dramatic changes in the behavior and physiology of cells. And in the case of cancer, those mutations can interfere with cellular cooperation, which leads to cheating. So what are some ways that cells in a multicellular body can cheat? Well, all of these sort of ways that cellular cooperation can break down. And really the problem, though, is that once those aspects of cheating arise, they are selected for by evolution among cells in the body. So, there are many levels on which evolution can operate, obviously, right? We have the organism level, we have the cellular level, you can even have, evolution operating within genomes on genes. Here, what we're talking about is selection that is happening among cells that are inside the body of one individual. And those cells that are able to acquire more resources, proliferate more quickly, avoid cell death, they have an evolutionary advantage. And so they expand in the population. And so this is sort of the essential, this is the core of what it means to take an evolutionary approach to cancer. And in terms of somatic evolution, is how those cells are gaining an evolutionary advantage within the body. All right. This is just another way of kind of visualizing what's going on. We have on the inside the foundations of multicellular cooperation, and then on the outside we have the sort of breakdowns of that multicellular cooperation that can happen in cancer. And now I'm adding one more layer on the outside of this. These are the hallmarks of cancer, according to Hanahan and Weinberg, who really put together what is considered the authoritative list of the phenotypes of cancer. And what we see is that they map on largely to the breakdown in these foundations of multicellular cooperation. There are a couple of exceptions that are kind of interesting. So it turns out that there actually is no hallmark in this sort of standard Hanahan and Weinberg framework. there's no hallmark having to do with dysregulated differentiation or breakdown of division of labor. So we've suggested that this might actually be a missing hallmark. In fact, if you look at how do pathologists look at cancer, they look to dysregulated differentiation as one of the main things that they use to assess if something is cancerous or not. So, and just for a little more perspective, the hallmarks of cancer framework really came from cell biology. And so it's interesting that the pathologists who are looking more at the tissue level, they focus on dysregulated differentiation, which wasn't part of the sort of, framework of hallmarks that came from the cellular perspective. But this framework that we're taking of thinking of cancer as a breakdown of cellular cooperation points us to a much broader picture, which captures both the hallmarks of cancer from the sort of cell biology perspective and potentially also some of these tissue level phenomena that pathologists focus on. So, so this suggests that there's something perhaps fundamental about this cellular cheating and evolutionary dynamics perspective on cancer that's capturing many different aspects of how cancer is approached, both from the cell biology perspective and the pathology perspective. Okay, so now that we've kind of talked about how cancer is a cellular cheater in multicellularity, I want to show you guys the premiere of a music video called Revenge of the Somatic. So this is based on a song that Baba Brinkman wrote, and I consulted with him on the science. He is, probably most of you know him, but he is the only peer-reviewed rap artist out there, I think, and he writes raps about evolution and medicine and climate change and lots of topics in science. So if I can make this work now, let me try to show it to you. All right, can everybody see my screen?
Yeah.
Uh-oh.
A couple 100,000 puffs of tobacco smoke, and I just wasn't as open to apoptosis. That's a bad prognosis. I was hit with
All right, let me see if I can go back to my presentation now. Actually, why don't we take a moment if anybody has any questions or anything. Uh-oh. Yeah, sorry. Autoplay, YouTube autoplay.
If anyone has any questions, you can use the raise hand function in Zoom or post them into the chat. Okay.
Otherwise, I'll just keep going.
I had a quick question, Athena.
Yeah.
I don't know if it's a good time to ask it or not, but
Sure.
you know, one of the things that occurs to me is, of course, variation across species, because I'm always thinking, you know, as a comparative biologist.
Yeah.
And of course, I'm familiar with Peto's paradox. Maybe people here would like to hear your perspective on Peto's paradox. But when, you know, when I look at the association between body size and cancer rate, for example, in the context of Peto's paradox, sure, there's a flat line there, but there's still a lot of variation around that line. And I was just wondering what comes after, you know, Peto's paradox. What is the next kind of paradigm that we could have for thinking about that variation across species?
Great question.
And in particular, whether you're five, I don't know what you're calling them, I couldn't quite catch it, but the hallmarks that you have, you know, sort of like the Hanahan and Weinberg, those 5 hallmarks, if that helps us understand what some of the variables might be that explain variation in cancer.
Yeah, well, so I'll give just a 1 or 2 minute explanation of what Peto's paradox is, and then we'll jump into that. I think it's a great question. So, so basically, Peto's paradox is this observation that cancer rates or cancer incidence is pretty flat across species. And if you would predict that, you know, cancer is simply a matter of cells mutating, and there are no differences across species in the biology of that, then what you would expect is larger and longer lived organisms should be more susceptible to cancer because they have more cells that are around for longer. So just more opportunities for cancer mutations to arise. So that's, you know, if there were no differences in the underlying biology. But there are differences in the underlying biology, and that is part of why this sort of overall effect of, you know, either, you know, no relationship or sometimes even a decreasing relationship with body size is observed. So we have a huge database, comparative oncology database that I've been developing with Amy Body and Carlo Meli for almost a decade now, where we have hundreds of thousands of pathology records from zoos and veterinary clinics, and we're able to actually look at what factors are associated with cancer risk in terms of not just body size and lifespan, but other life history factors. So, so to your question, Charlie, of, you know, sort of what's next after just, okay, you know, Peto's paradox exists, like we've, you know, shown that, it's pretty clear. Then the question is, well, what are the other factors that predict where species lie on these elements of cancer risk? And we're at the moment focusing in on things like litter size, age of first reproduction, some parental investment variables, so that we can try to see, you know, to what extent are these life history factors influencing cancer risk? And I think that there's a lot of, we're seeing a lot of promising results now. It's not at the point where we sort of have a, you know, one clear story about what's going on. And there's some things that are kind of puzzling in our data that we're trying to sort out. But it does look like on the whole that there are signals associated with life history variables like, you know, litter size, age of first reproduction, and parental investment and things like that.
That's great.
Yeah. Are there other questions now, or should I show some more of my slides? Looks like there's some questions.
There might be a related question here from Joe Alcock. Joe, do you want to ask your question related to that, or hold off till later?
See. Yeah. I was just wondering about the idea that organisms might expend effort to reduce cancer risk. And we know there's trade-offs between growth and, say, immune effort, depending on, say, the infectious disease burden. And I was wondering if anybody's attempted to look at trade-offs between growth or other life history traits and cancer risk, really in terms of the energetic costs, or if anybody's even attempted to look at or estimate those energetic costs, assuming that they cost something, as you mentioned.
Mm-hmm. Yeah, so I don't know of anybody who is looking at it from an energetics perspective. Amy Body and I, we did do a model with Hannah Coco, where we essentially looked at the potential trade-offs between cancer defenses and reproductive competitiveness, where, you know, reproductive competitiveness might include, you know, larger body size, ornaments, you know, things that may make an organism more likely to get selected by the other sex. But that really was a theoretical model, as opposed to like actually looking at the energetics of it. One thing I do want to mention, though, in the sort of energetics realm, is that there are people who are working on cellular energetics in cancer, and there are people who work on organism level energetics in cancer. And there is very, very little crosstalk among them or between them, which I think is really disappointing, because, you know, we know that the cellular energetics are going to be affected by the organism level energetics, right? Because you have to have, you know, things coming into the system in order for cells to be able to use those resources. So I think that there's a lot of important work to be done really on connecting the organism level energetics with the cellular energetics, so that then, you know, these kinds of issues can be looked at in greater depth.
Thanks.
Great, thank you. Looks like we have a couple more questions. Did you want to get back to your slides, or do you want to keep going with the questions?
I'm happy to kind of go into discussion mode now, and if questions come up where I have relevant slides, I'll pull them up. One thing I didn't talk about yet is evolutionary approaches to therapy. I'm happy to chat about that if that's something people want to just talk about informally, or show some of those slides later on. I'm happy to, but I'm happy to take some more questions now, and we can see where that goes.
Okay, awesome. Well, next up for questions, we had Cynthia Beal with a question. Cynthia, you can unmute yourself and go ahead.
Hi, Athena. This is Cynthia.
Hi, Cynthia.
Well, I asked a couple of questions on the chat box, and you have inadvertently answered some of them. I was asking about the extent to which your comparative database might have information on whether there's species variation in the extent of involvement of the oxygen sensing system in response to solid tumors, which enables them to grow bigger. That's an example of influencing the extracellular environment. And also related to that is the switch to glycolysis.
Sure. Yeah. So in tumors, often there's a switch to metabolism that relies more on glucose and less on oxygen. And it's thought that essentially what happens, you know, as the tumor is growing, it actually is creating conditions where oxygen can't get to the cells, right? Because in order for oxygen to get to cells, you have to have blood flow. And that's how the oxygen gets to the cells. But as a tumor is growing, it is creating conditions where there's less and less oxygen. And so the cells that survive are going to be ones that are more likely to be relying on glucose. But there's also, in addition to, you know, potential for selection there's also a shift that happens in the state of the cells. So in low oxygen conditions, that actually can shift the shift the metabolism towards glycolysis as well. So you have both kind of evolutionary dynamics, and you have kind of these, quote, behavioral adaptations of cells to their environments that can lead to these changes that can then actually spur on cancer even further. So one of the things about about glycolysis is, it relies on glucose rather than oxygen. It produces way more waste products, which are acidic and can actually damage the extracellular environment. And then that creates a situation where it's much easier then for cells to invade, because the extracellular environment has been broken down by that acid. So I think that, these changes in cell metabolism, what cells are producing, what they're utilizing in their environment, are extremely important for understanding the transition to invasion and metastasis. That is really the most clinically important issue in cancer, and it's something that is surprisingly poorly understood still. Why metastasis happens, why invasion happens, and it's not just a matter of there's no mutation for metastasis or invasion. It is a really hard to understand process that involves evolutionary and ecological dynamics that are really complex.
Thank you.
My pleasure.
Thank you. So it looks like we have one more raised hand, and then we have quite a few comments in the chat about people wanting to hear about the evolutionary approaches to therapy. So let's take the one raised hand question from Brooks Roby. Brooks, you're unmuted now, so you can go ahead. Well, hold on. There you go. Okay, go ahead, Brooks.
So I was going to speak to the answer to Joe's question, where Athena was talking about the discreteness of looking at it at the cellular level and at the organismal level. And I'm not so sure that that's really true anymore. I mean, there's data going back, to Eisenberg's work and Winehouse's work back in the 70s that the cellular functions clearly don't operate in vacuo. Cells are open systems, so they depend on the availability of nutrients and so forth. And so the Cori cycle, gets ramped up in patients with cancer, so that all that lactate that's being produced by this increased glycolysis is then converted back into other metabolizable substances and so forth. But there's a lot of data now that is also pointing to the ability of cancer cells to look at a broader repertoire of energy sources. But I hesitate to use the term energy, because by restricting the discussion to energy, we ignore the fact that metabolism is serving amphibolic functions. It's serving both anabolism as well as catabolism. And so not only do you have to have energy to do these things, you also have to convert the raw materials that are available to you into the substances to make more nucleic acids, to make more proteins, to make more membranes. And so by separating these things out, I think we've oversimplified a lot of these processes. But the cellular data seeks quite well into the physiologic interdependence data as well too. And I like to think of cancer cells as kind of being billy goats. It's not that they give up their ability to eat, specific types of nutrients, is that they expand their repertoire. And that glycolysis provides a platform for diversion into all these different anabolic pathways as well. So it's not just ramping up glycolysis. What you're doing is you're diverting through the hexose monophosphate pathway. You're actually coupling to mitochondrial metabolism, whereas Warburg originally thought that there was a damping down, but we know that oxidative metabolism, continues and actually supplies a lot of materials that go through that. So I guess what I'm doing is just pushing back a little bit, because I don't think that those things are as disconnected as was represented. I think that there's a lot of marriage between those things. And I think that there's a lot of work on those fronts that's germane to these.
Thanks for those comments. Yeah, I'll clarify. I'm not saying that they're disconnected fundamentally at all. I've just observed, you know, when I've been at cancer meetings, you know, I'll ask a question to somebody who is, you know, doing work on cell level metabolism about the relevance to organism level metabolism, and they look at me like I've, you know, just asked them a question about something that is so completely irrelevant to their work that they can't believe I asked it. So I think you're absolutely right that fundamentally these are connected, and they need to be connected in order for us to make progress on truly understanding them. I think there's just still some work to go in terms of probably the sociology of science, that a lot of these are still, you know, considered different disciplines. So, you know, I think there's maybe not an incentive structure in science for people to be collaborating in ways that allow for us to push forward in the way that I think would be best for the science.
Great, thanks for elaborating on that. So I think let's get into some of the evolutionary approaches to therapy. And Rob Dunn had asked, what do you think cancer treatment and management will look like in 50 years? Maybe you can kind of segue into kind of some future perspectives too.
Perfect, great. I'm gonna share my screen again here. Okay, I will briefly show you some photos of what cancer looks like across life, and then we'll segue into the material about cancer treatment from an evolutionary perspective. So here are just some examples. Actually, you know what? You guys are seeing my screen share in the wrong way. Let me fix that. There's all these. Here we go. All right, can everybody see it properly now? Okay, good. Yep. Excellent. All right, so if we look at, oh, it looks like I lost some of the slides. Here we are. Okay, so sorry, technical problems. Okay, all right. So if we look across vertebrates, we see that there are cancer-like forms, not just in animals, but across vertebrates, including so we have here a photo from a starfish on the top. You can see there's a little cluster of melanocytes. And also in clams, in other mollusks, we see cancer. Cnidaria, so this is coral, hydra, other cnidaria. We see both tumors and other things that are cancer-like, like loss of differentiation. In fungi, we see loss of differentiation and inappropriate tissues in inappropriate places, which is another cancer-like form. And then here, now we're coming back to our fasciation. So abnormal proliferations in plants because of meristems that have sort of grown into these bands. Interestingly, there's also we found one observation of a metastatic-like growth in a plant that did not have bacteria associated with it or anything. So that suggests that there might also be things that are more like invasion and metastasis in plants as well, although more work needs to be done really to see whether that's the case. All right, I'm going to skip ahead a little bit here. Here, there was a question about TP50 or about Peto's paradox. So I'll just mention this real quick that, you know, if you have a very large body, long lifespan, then you need more mechanisms for suppressing cancer. And our team has seen that elephants actually have extra copies of the tumor suppressor gene TP53. And other groups have found that these TP53 copy numbers increased over evolutionary time, basically with body size. So it looks like body size kind of increased first, and then copy numbers of TP53 may have followed. And the way that this was done is sort of comparing with woolly mammoth and mastodon genomes with modern day African elephants in order to look at how TP53 may have evolved with body size. Our group right now is doing some more in-depth work looking at different species of elephants and TP53, and the story is probably a little bit more complicated than just TP53, more TP53 larger body size based on that work that's ongoing. But nevertheless, it does seem like at first pass, having these cancer suppression systems like TP53, but not limited to TP53, is one way that organisms help to kind of keep cancer defenses up if they are long-lived and larger. And this is not just in elephants. Our team has also looked at giant cetaceans, so whales, dolphins, and has seen that there is positive selection for genes that are involved in making sure that the cell cycle is moving the way that it should, that cells are proliferating appropriately. So this is not just limited to elephants. It's not just limited to TP53. This is a broader phenomenon that you sort of need to have these systems in place for detecting and suppressing cellular cheating in order to grow large. All right. So what is the future of cancer? I think that was one of the ways that the question was worded, or sort of where are we going with cancer? And so I want to take a few minutes to talk about evolutionarily informed treatments and how they can potentially help us to control cancer. So there are a lot of ways that you can actually, instead of trying to, so I'll back up for a moment. The standard approach really in cancer treatment has been, how do you kill the most cancer cells? And we know from an evolutionarily informed perspective that if you try to destroy a large portion of the population, and you do that in any way that's systematic, you actually select for resistant cells. So this is, you know, what Baba Brinkman was kind of referring to in the music video about, you know, sort of using chemotherapy, right, and deading the weakest, and then the fitness of the remaining cells is much higher because they can actually expand into those niches that were where the cells that died are no longer. And he uses the term competitive release, and this is a term from ecology, which is basically if you take away competitors, then a population can expand dramatically. And so essentially that's what goes on when you use a high dose chemotherapy. And if you are not able to completely destroy every cancer cell, which is very, very hard to do. So if you're not able to do that, then the cells that remain are the cells that are resistant. So that's a problem, because then you cannot use the same therapy in the future to keep those tumors under control. So what are some other strategies that we could take? Well, if we come from this perspective of evolutionary biology and cooperation theory, then there are some new strategies that this suggests. So one is, you know, how do we support the cooperation of normal cells? And this is maybe more of a prevention issue, right? How do you keep cells cooperating with each other in the way that allows them to help our body function, help us be healthy, and make it so that cancer doesn't arise? Then there's a question of, well, how can you potentially support the cheater detection systems of the body? So how do you make it so genes like TP53 can function normally? How do you make it so that the immune system is able to detect cancer? So a lot of immunotherapies really kind of fit in this realm of supporting or rebooting cheater detection systems that are there. There's also the possibility of disrupting cooperation of cancer cells. So one thing I didn't really talk about today, because it adds another layer of complexity, but it is extremely important, is that cancer cells can actually cooperate with one another. They can produce public goods for each other. They can protect each other from the immune system. They can facilitate each other's invasion into tissues. So there's a lot of cooperation that is happening among cancer cells that is most likely extremely important, especially for those phases of invasion and metastasis that are clinically most important and currently the most mysterious still for oncologists and cancer biologists. So, how can we disrupt that cooperation of cancer cells? And then really simple one is slowing down evolution in the body. So, essentially, if you could just slow down the rate of evolution happening among cells by half, then most people would die of something else before they die of cancer. So that's a really interesting kind of out of the box sort of way of thinking about, how we might reduce the risk of cancer. But there's some very simple ways to actually do it. Like there's some results suggesting that using anti-inflammatory drugs can slow progression. And then there's minimizing the evolution of resistance, which I'll come back to in a couple of slides, because there's, I think, a really exciting approach to therapy that uses that. So here's one set of strategies that Bob Gatenby and Gunther Johnson and I talked about in PNAS, which is that one way of approaching cancer that kind of takes into account an evolutionary perspective and also challenges the existing way of operating is to focus much more on how do you protect the patient while limiting the tumor, as opposed to focusing on how do you kill the tumor. And some strategies that actually have been used in infectious disease treatment fairly successfully are to think of, well, what are the limiting resources, and can you limit those in a way that makes it so that the tumor can't grow as effectively. You can think about how to disrupt the cooperation that's happening among the cancer cells. We talked about that already. And you can also focus on preventing damage to the host. So rather than trying to sort of destroy the cancer cells per se, you look at, well, what are the ways that the cancer cells damage the host, and then how can you protect the host's tissues from that damage? Another exciting one, so this refers back to this issue of slowing down the rate of evolution. So essentially what happens often is that you can get cancers that have a very high mutation rate. And when the mutation rate is high, then that makes for more variation, which means that selection has more to operate on, and evolution can therefore happen more quickly. And so this is a really interesting study that was done where they looked at the rate of, or the risk of progression from Barrett's esophagus to cancer in people who were taking non-steroidal anti-inflammatories and people who were not. And now there's one caveat, which is people were on or off of NSAIDs. This was not a study that was done specifically to look at this question. This was using the existing variation that was there, because some people were on NSAIDs for treatment. I believe it was their blood pressure, their hearts. So they were looking at basically who was taking a baby aspirin every day and who wasn't. Then they had them go on or off depending on if they had been on or off before, so that they were able to look across individuals, but they weren't able to control for time entirely. But nevertheless, what see is a pretty dramatic change in the mutation rate. So if people were, first on NSAIDs and then went off NSAIDs, that there was a much higher rate of mutation during the time period that they were off NSAIDs. And if they were on NSAIDs, there was a lower risk of progression from Barrett's esophagus to cancer. All right, and now on to, I think, probably the most exciting of the approaches, evolutionary approaches to cancer therapy, which is adaptive therapy. And the thing about adaptive therapy that's really fascinating is that it's able to actually control tumors without really high dose treatment. So essentially it aims to treat just enough to keep the tumor under control without selecting for therapeutic resistance. And this makes it so that you can continue using the same drug when the tumor starts growing again. So here's results from a trial from a few years ago done by Zhang and Gatenby, where they had people who had prostate resistant or had castration resistant prostate cancer. And what they found was that when they had patients on adaptive therapy, they survived for years longer, basically, than the contemporaneous cohort that was not on the adaptive therapy protocol and was instead on a standard protocol. So this is very promising. Now it's a contemporaneous cohort study as opposed to a randomly assigned study, but the results are really dramatic. So if you look at the top, you see there was only one patient that progressed in the adaptive therapy trial. If you look at the bottom, all those X's are the patients who progressed. So it's a very, very dramatic difference. So it's promising, and right now there are a lot of efforts to bring adaptive therapy into clinical trials more. It's a challenge because there is there's nobody who can financially benefit from it at this point, which makes it a lot harder to get clinical trials off the ground. So I know Bob Gatenby is struggling with that at Moffitt, and we're struggling with that here now. We're trying to start a trial on adaptive therapy with breast cancer as well. So, I think there's some issues that have to kind of be worked out in terms of the financial incentives and, how do we actually take some of these really promising evolutionarily informed approaches and get them into the clinic and, get them into the trials that will allow for them to become more disseminated. And I just want to end with saying that I think, really the future of cancer is figuring out how do we live with it rather than dying of it? How do we keep tumors under control so that we, go through our lives with them, and, maybe we die at an age where we would have passed away anyway, and maybe we die with cancer instead of dying of it, right? Maybe it's there, but it is not the cause of death. And I think this is something that, we can also sort of see that multicellular life has coexisted with cancer since the beginning. And this is just kind of bringing that into our way of thinking about cancer more strongly. So I'm gonna leave you with a quote from Charles Darwin. There's grandeur in this view of life, from so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved. And we got the name for our crested cactus garden from this quote from Darwin. It is endless forms, most beautiful. So that is my talk, and thank you guys all so much for your attention. And I know that we're at time, but I'm happy to stay on for a few minutes and take questions if people have any. So.
Okay, thank you very much, Athena. That was great. Thank you.
My pleasure. Thank you.
It was really wonderful. We are out of time, and so, oh, and you put your, I see you have the link up here again for the code. I'm looking forward to buying a copy of your book. Congratulations on its publication and your promotion. That's great news. And thanks very much. We have a really nice lineup of speakers all the way into almost into July at this point. Next week we have Sean Carroll. We also have, after that, Jenny Tung and some of her colleagues talking about their new science paper that was out on social determinants of health. I think that's going to be a great conversation. So please tune into that after Sean Carroll's talk. After that, we have Bernie Crespi, and then Verena Schunemann from Zurich, who will talk about her work. So we have at least 4 people, and maybe Meredith is going to tell me I've forgotten a couple others, but those are the ones that I have in my mind. And if you want to stay abreast of things, please sign up for at the TriSem listserv for getting announcements about these events. So thank you very much, Athena, and I'll turn things over to Meredith. And it's fine with me if if it's okay with Meredith for people to stay on a little bit longer for questions. But Meredith, anything else you want to share?
Nope, I think you've covered everything. I'm happy to stay on for longer if people wanted to chat. I know we're officially over time, so do feel free to leave if you need to go. But if people wanted to chat, feel free to raise your hand and I will call on people and we'll keep going until Athena's tired.
I can stay on for about 10 minutes or so, 10 more minutes.
So it looks like we do have a hand raised from Joe Graves. Joe, you want to go ahead?
You're muted, Joe.
We'll get you unmuted. Okay, you're unmuted now, Joe.
I'm unmuted. First, thanks. Great talk, Athena. And I just bought the book, looked through the table of contents, but I haven't had a chance to really go through the section where you talk about cancer from womb to tomb. Now, you know that I'm a great champion of the Medawar-Williams-Hamilton evolutionary theory of aging. And one of the most obvious things about cancer in metazoans is it's strongly age-related in both prevalence and mortality rates, which are well explained by the Medawar theory of aging. In other words, after net future expected reproduction is 0, there's no longer any selection for cellular cohesion. So all of the genes which are beneficial early in life and which manage to allow that to occur sweep through populations, as I was attempting to explain in the chat. But then afterwards, they're antagonistic pleiotropy, meaning they start doing what they did when they were not cellular cooperative, which is what I think your book explains really well. So at the end of the day, while you're reproductive, it's beneficial to cooperate. And when you're not reproductive anymore, so even calling it cheating, I think is sort of irrelevant.
Yeah, very interesting point. So I think that there is certainly an interaction here between sort of, what happens to selection as we age, right? And after reproduction, certainly, that shadow of selection is much weaker. And then when you get to the point where you're not having much of an effect on the viability of your genes in any way, right? Then yes, you do have that weakening of selection. Now, there are a couple of kind of interesting details that come up though when we think about cancer in this. One is that it's not just a matter for humans of reproducing because we have really extensive parental investment. So we can enhance the viability of our offspring and of, the genes that they carry, right? By being alive and being functional for much longer. And so in species where you have a lot of parental investment, where you have cooperative breeding, where you have the capacity to sort of be continuing to enhance your inclusive fitness for a longer amount of time, then you can actually get selection, favoring cancer suppression mechanisms into, older age at least. So there's some hope for us. It's not just done, after we've completed our reproductive age. The other thing that I wanted to comment on, you bring up a really great point that, well, should we even think of it as cellular cheating if what happens is really just, a breakdown of things as we age? And I think that one reason to still use the cellular cheating framework is that the kinds of things that evolve inside the body, right? So you have all, 30 trillion cells, they're, cooperating, coordinating, making us function. The kinds of things that do take off, evolutionarily speaking, inside the body are situations often where cells are exploiting that cooperative system, where they are proliferating too much, where they're using resources too much. And so the actual phenotypes of the cells are consistent with cellular cheating. And yes, part of the reason, that they're evolving is because you're getting a breakdown of the, mechanisms that might be keeping them in check because of aging. But a lot of what emerges is consistent with cellular cheating.
So yeah, and that's great. And but I do want to point out that the theory for post-reproductive lifespan is abundant, and the number of experimental tests validating any of those theories is actually somewhat missing. So yes, there's a lot of arguments for why we live longer than we should, but nobody's actually demonstrated that those theories are correct.
Very interesting perspectives. Any other final questions before we officially close? All right, I'm not seeing any other raised hands. Oh, wait, let's see. Looks like we do have a comment. Is it possible that surgical resection of tumors already utilizes this principle, especially when compared to adaptive therapy? Is it also possible that surgery can be targeted one day?
Which principle?
So it says, I was thinking of reducing evolution, one of the strategies that you talked about.
Yes, yeah, definitely. So when you remove a tumor, you are dramatically reducing the population size. And population size is one of the factors that goes into how strong evolution, how strong natural selection can operate. So basically, if you take a bunch of the tumor out and you have less of the tumor to deal with, and you're reducing the population size. Now there can be a bit of a tension though between reducing the population size versus creating a situation where there is the possibility of competitive release. So the cells that are left may then have more resources, more opportunities, more space. So there's a trade-off there, and one action doesn't necessarily just have one unique effect on the evolutionary process inside the body in terms of how cells are evolving.
Great, thanks. So we have one last question. We'll make this the final question so you can get to your other commitments. But let's go to Kyle Summers, who has a hand raised. Kyle, you're unmuted. Go ahead.
Hi, I was interested in what you thought of the experiments on intermittent fasting by Walter Longo, which sort of tie into the idea of life history. I mean, he's arguing that when you go into a fasting state, our normal cells go into this kind of biochemical defensive crouch, if you will, and whereas cancer cells don't seem to. So he's had some really interesting results with mice, just showing that chemotherapy becomes much more effective in mice that are fasted. And I wonder if you'd seen those experiments and what your thoughts were in terms of an evolutionary perspective.
Yeah, I think those are really cool experiments. And I think they do relate to life history theory, but on the cellular level as opposed to the organismal level. So again, you know, we have these frameworks that are applying not just at the organism level, but also at what's going on cellularly. So I think, you know, it's quite likely that what you have is a situation where you know, cancer cells are sort of more fast life history, more optimized for, you know, gathering resources, growing, dividing, and not necessarily as much for surviving. But normal cells still have, you know, retained a lot of those capacities to survive. And so when you remove the resources, I think that that does dramatically change the environment for both the cancer cells and for the normal cells, and that it's possible that basically normal cells have still retained those abilities to survive when resources are restricted, while cancer cells have kind of, you know, already started down this pathway of fast life history evolution. So I think there's a lot of interesting dynamics having to do with life history theory at the cellular level that are just starting to be explored, and it's possible that this set of experiments inadvertently is actually getting at some of those questions, even though it didn't set out to.
Cool.
Great. Well, thank you so much for your talk today and for staying with us for an extra few minutes to chat a bit longer. This was great. And thank you to everyone for coming. I'm going to stop the recording now, and I'll send out a copy of the recording as well as a link to Revenge of the Somatic in a recap email soon.
Great. Thank you.